Oh, no. There we go. Okay, how did block 1 go? Happy to be back for block 2? I can feel the enthusiasm just radiating off you, yeah? Okay, so I won't see you a whole lot in block 2. You'll see me a bit today, but this morning we're covering some respiratory tract histology. So again, we've got a Padlet there for any questions that you may have as you go through. So our learning outcomes are quite straightforward. We're going to look at the different regions of the respiratory tract, looking at the sort of nasal cavity, larynx, trachea, bronchi, bronchioles, and alveoli, looking at the histological features and explaining how they relate to its physiological function. So I'm aware you've had 1 session so far, more looking at the physiology of stuff. So hopefully some of the sort of function I discussed today, you'll come across already or will come across at least later today or this week. There's a lot of slides, but it's just a lot of nice pictures, really. So it might not take the full session, but don't be put off by the fact there's about 50 slides or so. So functions of our respiratory system on the whole, it actually does quite a lot of things. So we have our gas exchange, so the exchange of oxygen and carbon dioxide between the blood and air. Defense, so when we inhale air, we may be inhaling sort of particulate matter or microbes, and we want to make sure we can protect the actual respiratory surface of our lungs from those particles and microbes, because that respiratory surface, as we'll see later, is very, very delicate. So we have a number of different mechanisms as we go through the respiratory system that are there to sort of provide that defense. It's involved in blood pH regulation. That's not something we're covering today, but you will cover that throughout the block of sort of how that works. It enhances venous return, so in terms of returning blood to the heart. So we know that a sort of cardiovascular and respiratory systems sort of work together in lots of ways. It's involved in the production of speech sounds and also olfaction, so the sense of smell and taste. So in terms of the functions we're going to come across today, gas exchange, obviously, we'll look at defense and how the mucosa, the epithelium, for example, are adapted to provide defense. The others we're not going to touch on so much. We'll touch a little bit on olfaction, but the others you'll come across sort of as you go throughout the module as well. But lots of different roles, not just gas exchange, and lots of ways in which the respiratory system works alongside the cardiovascular system. So in terms of organization of the respiratory system, you may have touched upon this already. We can look at it in 2 ways. So we can first look at it structurally. So this is looking at the structures that are outside of the thorax compared to those that are within the thorax. So we divide it by our upper respiratory tract, so our nasal cavity, pharynx, and larynx outside the thorax. And then our lower respiratory tract is everything from the trachea down. So the trachea branches into bronchi, bronchioles, and then all the kind of respiratory components of our alveolar ducts, alveolar sacs, alveoli, where that gas exchange takes place. So we can consider it structurally, but we can also consider it more functionally. So we can look at which regions are simply involved in the conduction of air from the atmosphere down into our lungs. So that's our nasal cavity, pharynx, larynx, trachea, bronchi, bronchioles, down to what's known as terminal bronchioles. So no gas exchange is taking place in these structures, but they are there to help transport air down to then our respiratory zone, where we actually see that gas exchange taking place. So we can divide it structurally or functionally, depending on sort of what we're talking about today. We'll mainly sort of be referring to it by its functional divisions, so conducting zones and respiratory zones. So that's just to make you aware of what we mean when we say respiratory zones, for example, as we go through. Just a reminder, because this is a histology session, we're going to be looking at epithelium again. So just as a reminder of that, although I'm sure you're all familiar by now. So our epithelium is our continuous layer of cells that form the covering of body surfaces, lines body cavities, and hollow organs, and is a major tissue of glands. And as we know by now, they can be categorized by shape. So remember our different shapes. We have our cuboidal, so they're roughly as tall as they are wide. Our columnar, that are taller than they are wide, and our squamous, which are those very sort of flattened cells. They may not necessarily always look as neat a shape as that, but by sort of estimating its width versus height, you can sort of figure out which shape descriptor you would use. And we also have the number of layers. So remember, our simple epithelium is a single layer of cells. So they're a simple columnar, and a stratified epithelium where we have one or more layers of cells. So where it's something like this, a stratified cuboidal, we wouldn't typically see more than perhaps 2 or 3 layers. But as we've seen before with something like a stratified squamous, that could be 15 to 20 cell layers thick. We have our pseudostratified epithelium, which is going to be featuring very heavily in today's session. So that's where it is a single layer of cells, but because the nuclei of those cells sit at different levels, it gives the appearance of a stratification, even though it's not. So that pseudo refers to it being a fake stratification. And also, just to mention the other type as well as transitional epithelium, where you can see from sort of the base to the most superficial layers, the cell shapes look different. And as that tissue stretches, the cell shape changes as well. So we see that, for example, in the urinary bladder. But as we mentioned, that pseudostratified epithelium is something we're going to see quite a lot today. And we'll also be pointing out any special features, so cilia, for example, on the epithelium as well. So that was just a reminder to make sure we're all familiar with the terminology. As I said, we're going to see featured very heavily today a particular type of epithelium that is pseudostratified. So not throughout the entire respiratory system, but for a large portion of basically our nasal cavity down to our sort of bronchioles, we see what's typically referred to or called a respiratory type epithelium. Now that name doesn't tell us a lot about what type of epithelium it is. It's actually a pseudostratified ciliated columnar epithelium with goblet cells. So you can see why people abbreviate it just to say respiratory type epithelium. This type of epithelium isn't just found in the respiratory system, but it is the predominant location of this type of epithelium is in the respiratory system, hence it's given that name. But I think it's much more helpful to remember that very long name because that tells you a lot more about the epithelium. So we can see that even though it looks pseudostratified, so even though it looks like we've got multiple layers of cells, every cell there, even though maybe you can't tell, but every cell is making contact with the basement membrane, and so it is only a single layer of cells. Some of them will make contact with the basement membrane and also extend out to the luminal surface. So these tall columnar cells are making contact with the basement membrane, but also have this sort of apical surface facing the lumen. The basal cells at the bottom here will be sat on the basement membrane, but they don't extend all the way through the epithelial sort of thickness. So we can see that if we look through the name, we said pseudostratified, so it looks stratified, but it's not ciliated. So you can see that lovely sort of carpet fringe on those cells there, the cilia on the apical membrane. They're columnar, so although we can't really see, it's not as nice as the cartoony diagram, they are definitely taller than they are wide, so they're columnar cells. And we can see here we have some specialized to secrete mucins, which when mixed with water will make our mucus. So we have those goblet cells there. And as I said, we'll see this throughout many of the different parts of the respiratory tract. I said not all of it, and there will be some little sort of like patches as we go along where we see a different type of epithelium for particular reasons. But we'll see this respiratory type epithelium in our nasal cavity, or the large majority of the nasal cavity, at least the paranasal sinuses and nasopharynx, false vocal folds, trachea, and bronchi. And then as we get into the bronchioles, it does transition as we go along to a different type of epithelium. But this is a predominant type of epithelium in the respiratory system, and it's particularly important with regards to respiratory defense. So if we just take a look at a cartoony diagram of that so we can see it a little bit easier on that. So I should say most of the stains we look at are this H&E stain, so hematoxylin and eosin, simply stains the nuclei purple and everything else pink. So oftentimes, unless it's something obvious like this, like a big goblet cell, we often can't distinguish very easily between different cell types unless we do some kind of special staining. So I'll point out some of those as we go through. But for now, we'll look at a very cartoony, very obvious diagram where everything's nice and color-coded just to explain some of the different cell types that we're seeing there. So we, of course, have those ciliated cells, so those columnar cells. as we sort of go through into the deeper parts of the respiratory tree, we will see that they go from a more columnar shape and become shorter, almost cuboidal, as we get to sort of peripheral bronchial branches into the bronchioles. But you know, in the sort of more superior parts of the airway, they do have that very columnar shape. As I said, we have our goblet cells, so they're secreting those mucins to create a protective mucus layer. And these two cell types, so ciliated cells and the mucus produced by those goblet cells, work together to create what's known as the mucociliary escalator. So what is that? Well, that is part of our respiratory defense. So effectively what we have is this mucus layer sitting on top of that pseudostratified ciliated columnar epithelium with goblet cells, and it'll capture inhaled particulate matter microbes. So for example, here you can see those little brown dots are just examples of inhaled particulate matter. So they get captured in that mucus, and then those cilia, so remember cilia are motile projections, they'll waft and they'll move that mucus, which has got that captured particulate matter or microorganisms, they'll move it along. So the ultimate goal is to move it to the pharynx. So if we're talking about the nasal cavity, it'll be moving it down to the pharynx, for example. If we're talking about sort of lower airways, the trachea, for example, it wafts it up. Once it's in the pharynx, we can then swallow it, and our stomach acid can deal with whatever it is that any microorganisms or particulate matter that we've inhaled, or perhaps not in polite company, you can spit it out. So it's a way of the body clearing itself of inhaled particulate matter. It's not the only respiratory defense mechanism we have, and we'll see as we go through. Before we even get to this point, what do we have in our noses? We've got hairs. Some of us more prominently than others. Some of us have to, I'm not saying me, but some people have to, you know, go with a little trimmer perhaps. But those respiratory hairs are really important. We don't want to get rid of them because they can capture larger particles. Any large particles that get past that will have this mucociliary escalator in the nasal cavity down through the trachea into the bronchi. And then when we get into those lower airways where we transition away from this epithelial type, we have other respiratory defense mechanisms down in sort of the alveoli to protect those delicate respiratory surfaces. And ideally, by the time we get down to our alveoli, hopefully most of this stuff has been captured and isn't going to get to that point. You can see this in action really well if you ever stay somewhere that has like a coal fire. So I lived somewhere once that had a coal fire, which meant every so often I had to clean out all of the ash and load up fresh coal. And then some time later, when I blow my nose, it would all be a lovely shade of like black that would come out because of all the sort of particulate matter that had been captured. So nice to kind of see it in action. And we can see here on an actual histology image. So there's our respiratory type epithelium. That's that mucus, and all these little dots are all different sort of trapped dust particles and particulate matter doing its job, capturing all that, preventing it from getting down into lower airways. Sometimes when we prepare tissues for histological sort of viewing, so we have to sort of process it, we have to take it through different chemical solutions, we have to stain it. It introduces what's known as artifacts. So that is, the tissue ends up not quite appearing the way it does in life, in our bodies. So the artifact that tends to be introduced here is this gap. So you can just about make out the cilia here, the little sort of fringe-like cilia, but the mucus is all the way over there. There's a gap between the two. Well, those cilia need to waft the mucus. They're not going to be able to do that if there's a big gap. This is an artifact. So it's not like that in life. It's like this in life. The cilia can actually waft that mucus. There's not this gap. This is just introduced because of the processing the tissue's gone through. It's important to bear in mind when you look at tissues if there's any common artifacts that can be introduced into that tissue to make sure that you're not kind of misinterpreting what's going on. And if we use a scanning electron micrograph to actually look at the surface of those cells, I just think this is a really nice picture. It kind of, to me, looks like a field of grass or something.
It's a reef.
A reef, a coral reef maybe. But that's all of those cilia, and that's kind of protrusions at the top of the goblet cells. It's just, we very rarely look at things with a scanning electron micrograph, you know, in any kind of routine practice because it's very expensive. but it's just quite nice to actually see what these structures look like closer. I always find that quite a calming image to look at. So going back, we don't only have those ciliated columnar epithelium, we don't only have goblet cells, we've got some other cell types here. We've got our basal cells, so as I said, they will sit on the basement membrane, but they're not projecting all the way to the luminal surface up here, so sort of smaller cells that sit at the basement membrane. And there are multipotent stem cells, so they're involved in regenerating and differentiating to replace lost epithelial cells. We also have another cell type here called club cells, also known as bronchial exocrine cells. So these are found more predominantly in the smaller airways and the bronchioles. So we see increasing numbers of them as we go into those smaller airways. So as we'll see in a moment and as we go through, we don't sort of have a consistent number of different types of cells as we go through the respiratory system. We'll see more goblet cells in sort of the more superficial regions. As we then descend into the bronchioles, they disappear, but then we start to see more club cells there instead. As I said, we couldn't necessarily distinguish those club cells on the previous H&E because it's just too standard a stain to kind of differentiate different cell types, but we can use different special staining. So it's known as immunostaining or immunohistochemistry staining, where we can look for specific markers that are only expressed or specific substances that are only produced by those cells to highlight them. And the key with this is just basically wherever it's brown, that's telling you where it is. Everything else is blue. So everywhere brown there. So this is all of our epithelium here. All those brown staining cells are those club cells we just mentioned. So it says here, this is a bronchial sample. So we start to see more of them as we go down into those lower airways. Club cells secrete a protective substance known as club cell secretory protein, and this helps to do things like detoxify harmful substances which have been inhaled. The club cells can also act as stem cells as well to help regenerate the epithelium. You may see, although hopefully in modern textbooks they don't refer to it by this, you may see them referred to as Clara cells. That's because the individual who originally identified them was somebody called Max Clara. Unfortunately, he was a member of the Nazi Party, and he did a lot of his work on victims of the Third Reich. So back in 2013, some of the respiratory system journals got together and said, we don't want to continue honoring this guy by continuing to call these cells Clara cells. So they got together and decided to rename them. So hopefully now in most places you'll see them referred to as club cells, but if you do see anything referring to Clara cells, it's the same thing. That's just an older name for them. But for obvious reasons, we try not to use that name anymore. Now, in most textbooks, they may be the majority of cell types that are mentioned, but as with a lot of science, what's written in sort of textbooks, etc., is kind of just our understanding at that point in time, and our understanding of how the body works in pretty much every regard is constantly evolving. So that is the traditional picture of the airway epithelium, that respiratory type epithelium. It's not necessarily as straightforward as that. So further work has been done to look at the respiratory epithelium and try to really identify what cell types are there, what do they do. And that's really important, not just, so we can write a more detailed textbook, because the more we know about the different cell types that are there, the better we can address treatment of respiratory diseases if we know perhaps what particular cell type is deficient or defected or affected in that particular disease. So I'm not going to go too much into the research side of things, but just to kind of point out how sort of clinical practice and research can come together. When looking at the respiratory epithelium, we need samples of it, right? I don't know about you, but I'm not putting my hand up to offer for somebody to come in and kind of take samples of my respiratory system. It's not a very nice process, perhaps. So when the researchers did some of this work to try and distinguish the different sort of cell populations, they took samples from clinical samples. So patients who were going in for diagnostic procedures, for example, having bronchial lavages, for example, having brushings taken for some kind of diagnostic purposes. Those samples, obviously with consent, but those samples were then also used for research purposes. So it prevented, the patients were having that procedure anyway. The sample was there. If we could just use some of that, it kind of prevents any unnecessary sort of invasive procedures to gather samples. So obviously in that circumstance, we're talking about patients who maybe have a respiratory disorder, but it's useful to also have samples from healthy individuals to compare, because in different disorders, there may be variations in the type of cells present. So there's biobanks that exist where researchers can access normal healthy tissue samples rather than having to get a person off the street to come in and take a sample from them. So they took all these samples and they performed something known as single cell RNA sequencing. So they took their sample, and in that sample, we're going to have lots of different cell types. Some we know already, some that maybe that we haven't been able to quite distinguish before. Maybe we thought we had all these 10 cells here. We thought they were all the same type, but maybe we've actually got different subpopulations of cells in there. We've just not been able to discern them yet. So within that sample, we'll have a variety of different cell types. We do some sorting known as fluorescence activated cell sorting. So we know on this particular cell, it has these particular markers, but on this cell, it has these markers, which are different. So we can look for those markers and separate out the cells. And then we can do single cell sequencing. So taking those cells we separated out, we can look to see what is their actual gene expression profile. So we know that all cells have the same DNA. All cells have the same genes, but different cells will express those genes to different amounts. Some will express loads of a particular gene, some will express very little of it, and that kind of distinguishes one cell type from the next. So they look for the RNA, so the actual expression of those genes. They look for the RNA because that's telling you that the gene is being expressed. And then they can quantify to what extent that RNA is being expressed. And they come up with what's known as expression profiles. So for each gene, there'll be a corresponding color to say, is that being expressed a lot, a bit, not very much, not at all? And they can do that for a whole range of genes for each cell. And then they can come up with something like this, where basically they can discern subpopulations that they didn't know before. So for example, in this region here, this was cell type 1, so we thought they were all the same cells, but you can actually see there's two very distinct subpopulations forming. So before we thought all of these cells were the same cell, actually they might be very similar, but they look actually like they might be quite distinct subtypes of the cell. So I'm not going to test you on that, but that's just to tell you how we kind of figure these things out. And what we found out, that the picture looks more like this. There's more cell types there than we thought, doing different jobs. Some of them are perhaps, so for example, these deuterosomal cells, they're a subcluster precursor of cilia cells. So they may ultimately be sort of differentiating, for example, into ciliated cells. We can see sort of different stages of cell development. We can see neuroendocrine cells that perhaps we didn't know were there before. They might not particularly be in great number, for example. So in terms of pulmonary neuroendocrine cells, it's only about 1% of the respiratory epithelium is these cells. But it's important that we know this because, for example, neuroendocrine cells, they do things like stimulate immune and physiological effects. So they can cause, for example, local vasodilation. They're really important in the fetal lung to help with sort of directing growth and development of the fetal lung. We saw similarly neuroendocrine cells when we looked at the GI tract. So for example, the G cells secreting gastrin, for example. So, if we can understand more about the cell types that help direct physiological processes, if those processes are aberrant in disease, we can look at these cells. Are they acting aberrantly? Can we then target them to help with this treatment of this disease? They discern something known as ionocytes, so regulating ion transport. So notably the CFTR channel protein, so the cystic fibrosis transmembrane conductance regulator, I can never remember the full name, but it's basically the protein that is involved with transporting chloride ions into the lumen. Water then follows, and that helps make sure that the mucus we just spoke about is nice and movable, so the cilia can move it along. if that particular channel protein is defective and we don't see chloride ions moved out into the lumen, that mucus is thick and it's sticky, it can't be moved along. Any bacteria that's caught gets to stay there, it causes infection, and that's what happens in cystic fibrosis. So by finding out actually what cell type is predominantly performing this function, we can target it better. We can come up with drugs or treatments that specifically target this cell to help treat things like cystic fibrosis. So it's not just about finding out about different cell types for the sake of it. It can help then direct treatment, direct new sort of drug therapies with more specificity, because we know exactly which type of cell is deficient, how it's deficient, and then try to address that. We also found something, I say we, I didn't do it, the scientific community found something called tuft or brush cells. Don't really know much about them. They're thought to help evoke both positive and negative immune and neuronal responses, but people don't really know what they do, to be honest. They haven't really got that far in figuring out what they are. But maybe one day we'll figure out that they play a really crucial role and, you know, could be some kind of target for treatment of a common respiratory disorder. We just don't know. So you'll predominantly see, you know, the goblet cells, the ciliated cells, the basal cells, and the, what was the other cell? Cup cells, thank you. You'll commonly see those mentioned as the respiratory type epithelium cells, but keep in mind they're not the only type of cells. We see that there is a variety of other cells and probably more that will come through research as well. As we go through the respiratory tract system, so we'll start up in the nasal cavity, even though it's not shown on this picture, what we'll see is that just like we saw with the GI tract wall, we'll see kind of changing features as we go through. So with regards to the epithelium, sort of predominantly, although there's some little patches of variation as we go through, but predominantly we'll see that pseudostratified ciliated columnar epithelium with goblet cells. But as we progress down, we'll see that the goblet cells decrease in number. We'll see that those cells get shorter, more cuboidal. And then by the time we get into the alveoli, they're a squamous type of epithelium. So we see changing in the epithelium type. As I said, we see changes in the number of goblet cells. We see changes as to whether those cells are ciliated or not. So for the majority, they're ciliated, but by the time we get down to the respiratory zone, they're no longer ciliated. We'll see glands in the lamina propria and the submucosa, again, predominant in the sort of upper airways down through that sort of conducting zone. But as we get to the respiratory zone, we see none of those glands anymore. We'll see cartilage. So here we can see these sort of blue rings in certain regions. So the trachea sort of downwards. We'll see cartilage rings, cartilage plates, but again, disappearing by the time we get down to those lower airways. We'll see smooth muscle in the wall of the trachea, the bronchi, the bronchioles. That smooth muscle allows for some degree of sort of contraction of the airways to help regulate airflow. The degree of contraction is kind of influenced by how much cartilage there is. So in the trachea, as we'll see, we have these C-shaped rings of cartilage whose job it is to keep the trachea open. It's our one and only, you know, sort of access point for getting air into the body, so we don't want it to collapse. But as we get down to the lower airways, we'll see that the cartilage disappears, but we still have smooth muscle. And so what do you think that means for the lower airways? We've not got any cartilage, but we've still got smooth muscle. What will that smooth muscle potentially do? Contract, and with no cartilage to keep the airway open, can collapse. So in something like asthma, where the airways are irritated, the upper airways, well, we've got other issues. We have loads of mucus and stuff there as well. But in terms of the airway itself, the constriction as a result of the muscle contraction won't be a start because we have the cartilage holding it open. But in those lower airways where we have no cartilage, if that tissue is irritated, we have bronchoconstriction, we have contraction of that muscle, and the lower airways can effectively completely close off. And so even though we might be able to get some air in, if it's not getting to the respiratory zone, we're not seeing gas exchange. So the relative, hello?
like surfactant issues, would that be the same issue of them closing and not being able to open them, or would it be them staying open and it's closed?
We'll come to surfactant later on. So surfactant lines the alveoli, but it is very, very important for the alveoli staying open. Okay, but we'll come to that later on. So looking at sort of the relative proportions of cartilage and smooth muscle can mean different things in terms of what that smooth muscle can do in terms of narrowing the airways. We'll also see elastic fibers throughout the wall of the respiratory tract. So we are going to look at sort of nasal cavity pharynx, except for this one shows the trachea down. But probably didn't cover it in the other session, but this is because the sort of lungs themselves, the respiratory system, it's passive in terms of airflow. So in terms of moving air into our lungs through the respiratory tree down to our alveoli, we're relying on movement of muscles in the rib cage and in the neck. So as we inhale, we've got our intercostal muscles pulling the rib cage up and out. We've got, if we really want to take a deep breath, we've got our sternocleidomastoids and our scalenes to really sort of lift up the chest wall. And it's through sort of movement of the chest wall, the lungs move with it, that increases the size of the alveoli and allows air to flow in. And then exhalation is a passive process where we release the contraction of those muscles. The thorax moves back and in. Those elastic fibers are recoiling to reduce the size of the alveoli and air moves out. You're going to be covering more of that this afternoon, I think. That's just why elastic fibers are really important, because it's to do with the recoil of those tissues during exhalation to reduce the size of the alveoli, which then allows air to move out. But that will all make sense this afternoon. Okay, so starting at the top then, in our nasal cavity. So I know you've done all your anatomy before yesterday, if you remember that. So I'm not going to go through all of the anatomy now, just to point out that in our nasal cavity, we have a couple of different types of epithelium. So there we go. So around our sort of nostrils, our nasal vestibule here, so the exterior of our body is a keratinized, stratified squamous epithelium, our skin. Why is it stratified? Why has our skin got a stratified epithelium?
For abrasion.
For abrasion, yeah. So we're living in the world, there's lots of things that could sort of abrade. There's bacteria in the world, there's things that we want to protect against. So having that stratified squamous epithelium is largely protective. Why is it keratinized? What? Hello? Sort of, it's more about preventing water loss. So it helps prevent water loss from the body. As we sort of enter the nostrils, then the sort of immediate sort of interior is still that keratinized, stratified squamous epithelium. But then as we move more inwards, that turns into a non-keratinized, stratified squamous epithelium. And then in the large majority of the nasal cavity, we see that respiratory type epithelium. We see a sort of transition from that external skin epithelium to that respiratory epithelium. In the most superior part up here, we have a specially adapted type of epithelium, which creates our olfactory mucosa. We'll come back to in a minute, but that's involved in the sense of smell. And we can see that we have these shelves of bone, these turbinates, these shelves of bones, which are covered by that respiratory mucosa to increase the sort of surface area of the nasal cavity to help it do its job, which is filtering the air that's coming in. So we'll have the hairs in our nostrils doing that initial job, but then we'll have that mucociliary escalator help doing the job of capturing the particulate matter and removing it from the respiratory system. We also see warming of the air taking place here. So as we'll see in a moment, we have lots of blood vessels in the sort of mucosa. So the body temperature blood that's flowing through those helps to warm the incoming air. And we also have glands which are secreting a serous fluid, so a sort of watery fluid, which helps humidify the incoming air. So this warming and humidifying and filtering is all to help protect the lower airways from the air that's coming in that may be cold, that may be very dry, could be dehydrating, or could contain some kind of particulate matter that could be damaging. So as I said at the top, we've got some specialized type of mucosa. We'll come back to the olfactory mucosa, which is involved in olfaction or our sense of smell. So as we said, the large majority of the nasal cavity is that respiratory type epithelium, which is a lot easier to say than saying the full name, but our respiratory type epithelium, you can again see, you know, the cells can be quite tall. So that combined with the nuclei sitting on multiple levels really can fool you into thinking it's stratified when it's actually not. So, you know, if you were to trace every one of these cells down, they are sat on the basement membrane. You can see the beautiful cilia again. We can't really see many clear examples of goblet cells, but there will be some in there as well. If we look sort of in the underlying layers there in the lamina propria, it's rich in blood vessels, so labeled V there. So as I said, the warm body temperature blood flowing through these blood vessels will help to warm the incoming air. And we've also got seromucous glands as well. So some cells are secreting a more mucus, sort of thicker secretion, which again helps with that mucociliary escalator sort of function for capturing of particulate matter. But the serous cells, they're secreting a more watery secretion, which helps with the humidification of the incoming air. We have our paranasal sinuses. I know, again, you covered this in your anatomy. So we have those four paired structures, the frontal and maxillary, ethmoid and sphenoid, which have orifices emptying then into the nasal cavity. And these are also lined by the respiratory type epithelium, so also contribute to that warming and humidifying of the inspired air as well. Looking then at that specialized mucosa at the sort of superior part of the nasal cavity, that olfactory mucosa, it's quite different. It still has a pseudostratified appearance, so it still is a pseudostratified columnar epithelium, but the cell types we see there are quite different. So we have ciliated olfactory sensory cells, non-ciliated sustentacular or supportive cells, and we still have basal cells. So in the green down at the bottom there, that's our basal cells, so the ones that can regenerate to sort of replenish the epithelium. In pink, that's our sustentacular or supportive cells. So they provide sort of mechanical and metabolic support to the olfactory receptor cells. They also secrete odorant binding proteins as well. They don't have cilia themselves, these ones. They actually have short microvilli just to help with that secretory activity. The sort of interesting and I guess most important ones here, however, are those olfactory sensory cells, so the ones in yellow. So you can see they have a sort of bulging nuclei sitting quite centrally in the cell, and then they have sort of extensions progressing sort of in a bipolar sort of nature, so an extension coming up from the cell. So they have this dendritic process that extends towards the luminal surface, which then expands into this olfactory vesicle from which we have cilia extending. These aren't cilia in the sense of sort of being motile, but they have odorant receptor molecules on them. So they're to do with detecting sort of odorant molecules, so in terms of smell detection. And then extending downwards, we have these proximal processes which penetrate the basement membrane. So all these other cells, you know, sit on the basement membrane, but this region here penetrates it. And then the axons of all of these olfactory cells kind of come together to form the olfactory nerve, which then communicates with the olfactory bulb for the sense of smell in the brain, but then also communicates to other regions of the brain so that smell can be sort of connected to things like memory. So, you know, you can smell certain things and it can evoke a certain memory in you. So it communicates to other regions of the brain as well. We also have some glands here, so Bowman's glands, and they secrete a substance which acts as a solvent for the odorants to dissolve in, which then aids them in interacting with the odorant receptor molecules that are on these cilia of those olfactory cells. So looking histologically, you can see it looks a bit more cellular than perhaps the respiratory type epithelium we were just looking at. It looks like there's a lot more cells going on there. It still is a pseudostratification, even though it's a lot harder to tell there. It still is, you know, a single layer of cells. We tend to see the nuclei of those sustentacular or supportive cells sitting more at the sort of top of the epithelium, whereas then the olfactory receptor cell nuclei are found sort of underneath. But we can't really make out their sort of processes they have extending either way, again, because this is quite a standard stain, which doesn't really allow us to make out features. But again, we can see we've got some basal cells there, sort of at the bottom, and we can see those nice Bowman's glands secreting that solvent. Moving into our pharynx then, as you're aware, we've got sort of three regions to the pharynx. So we've got the nasopharynx, immediately sort of posterior to the nasal cavity. We've got the oropharynx, and then we've got the laryngopharynx. So because our nasopharynx is only a passageway for air, nothing else is hopefully entering our body through our nasal cavity, our nasopharynx has that respiratory type epithelium. That's the main job it's got to do. But when we get into our oropharynx and laryngopharynx, that's a common pathway for both air and food. And what's more important here is protection from the abrasive processes of swallowing food. So whilst we have that respiratory type epithelium in the nasopharynx, in the oro and laryngopharynx, we have a non-keratinized stratified squamous epithelium, because the predominant concern there is protection against the abrasive forces of swallowing a food bolus. So again, in our nasopharynx, it's a pseudostratified ciliated columnar epithelium, so respiratory type epithelium. We'll have seromucous glands we can't necessarily see in this picture, and the lamina propria and submucosa contributing to that mucus secretion and the sort of watery secretions that help humidify the air. But we'll also see in certain regions of the nasopharynx, and what we can see here is masses of lymphoid tissue. So lymphoid tissue is where we find immune cells, so B lymphocytes, T lymphocytes, maybe some macrophages, some plasma cells. So this is part of, again, the respiratory defense. So as well as having that mucociliary escalator, as well as having the nasal hairs, we have these masses of lymphoid tissue, basically at the opening of both the respiratory system, but also we'll see other structures for the digestive tract as well. So that things entering those orifices can kind of be sampled by the immune cells there, can kind of see what's going on. Is there anything coming in that shouldn't be that we need to launch an immune response to? So as well as the sort of masses of lymphoid tissue in the nasopharynx, we also have lymphoid tissue surrounding the openings of the eustachian tubes, which open, you can see here. We've got the palatine tonsils and lingual tonsils that we've mentioned before, and you can see they create this kind of this border of protection for anything entering via the nose or via the oral cavity. So air or food or anything coming into our body in that direction can kind of be sort of surveilled, so sort of sampled, looked at. Is there anything that's bad that we need to launch an immune response to? So the lymphoid tissue in the nasopharynx is sometimes referred to as the nasal tonsils or adenoids, all the same thing. All of this forms part of what we've mentioned before, which was MALT, so our mucosa-associated lymphoid tissue, which can be subdivided. So we saw in the GI tract, we had gut-associated lymphoid tissue, which was a branch of MALT. So we had our Peyer's patches, for example, in our ileum. Sometimes you may see the nasal lymphoid tissue referred to as NALT. So nasal associated lymphoid tissue, you tend to see that kind of subdivision described less often, but if you see NALT, they're referring to the nasal lymphoid tissue. But it's all under the umbrella of MALT. So we have, again, nasal hairs, we have that mucociliary escalator, but we also have this immune surveillance of anything entering the respiratory and GI tract as well. We then go down to our larynx. So again, I know you've covered this in your anatomy already. The main thing we're looking at here in terms of the respiratory system, because we're really looking at the histology, the epithelium, is our epiglottis. So we have that sort of cartilage structure that sort of works as a flap to help protect the airways and protect any food from entering the airways. So prevents food from going into the airways, prevents air from going into the digestive tract. So making sure everything goes down the right tube, essentially. So in breathing, we can see the epiglottis is sort of in an open position, allowing air to come into the trachea, but as food is swallowed, kind of acts as a little flap to sort of cover the top of the trachea to direct food down the esophagus, which sits posteriorly to the trachea, and make sure, hopefully, that it doesn't enter the trachea. We know the larynx has got a bunch of other sort of cartilage structures, but in terms of the sort of mucosa, it's the epiglottis that we're discussing here. So in terms of the epiglottis, we've got part of the epiglottis that's kind of exposed to the sort of oral cavity. So this side here, which we call the lingual surface. And then we have a surface that is exposed to sort of the pharynx side. So what we see is of the epiglottis, so just of that one little sort of flap itself, it actually has two different types of epithelium. So on the lingual side, so the lingual side, if we think about when we're swallowing food, when that little epiglottis folds over, it's the lingual surface that's going to have the food bolus passing along it, right? Passing past it. So the lingual surface is a non-keratinized stratified squamous epithelium because of that abrasive process. But the sort of underside, so this underside part here of the epiglottis, as it folds over, that's going to be basically in the environment of like the trachea. So it's not going to have that abrasive process occurring. So whilst on the lingual surface, we have a stratified squamous epithelium to protect against the abrasion of the food bolus, on the pharyngeal surface, we don't need to protect against that because it's not going to be rubbing up against any food. So on the pharyngeal surface, we have a respiratory type epithelium. It actually transitions about a third to a half of the way along. So on this sort of inferior portion, the pharyngeal surface of the epiglottis, we still see a stratified squamous epithelium up to around, this picture shows it, well, around a third of the way along. Some texts say around half the way along before it transitions to that respiratory type epithelium. So we see those two different types. If we take a sort of cross section of the epiglottis here, we can see this is our lingual surface at the bottom there, where we have that stratified squamous epithelium to protect against those sort of abrasive processes. Whereas on the pharyngeal side, it goes back to our respiratory type epithelium. And of course, the epiglottis itself is composed of an elastic cartilage sort of core. So you have two different types because the two different sort of sides of the epiglottis have different needs in terms of whether there's kind of that abrasion or not, whether it's being exposed to that passage of food or not. Then going down from our epiglottis, we get to our false and true vocal folds. So these mucosal folds, the vocal folds are not necessarily involved in normal sound production, but they are protective then to the delicate true vocal folds underneath, which do produce sound. And again, depending on their needs, we see different types of epithelium again. So we just discussed coming down from the sort of epiglottis here that we had the pharyngeal surface with the respiratory type epithelium. That continues and covers the false vocal folds. So we have the respiratory type epithelium. But then when we get to our true vocal cords here, we see a very brief switch back, that stratified squamous epithelium. Any ideas why? We shouldn't have any food here at this point. We're truly in the respiratory system now. So any ideas why the true vocal folds would need a stratified squamous epithelium?
Stress. Well,
What kind of stress? What's actually, say, when you're talking, what is making that sound? Vibrations. So as they're vibrating against each other and rubbing, it may not be as stark a kind of friction, you know, as a food bolus going down, you know, your esophagus or something, but that movement, that vibration of those true vocal folds, that is an abrasive process. And so they have a stratified squamous to help protect them against that. And then once we get past that region that's going to be vibrating and producing the sound, and we then start to transition into our trachea, again, switches back then to the respiratory type epithelium. So this is a particular region where we just see lots of kind of switching back and forth between these two epithelium types. But the crucial thing to remember is where we're going to see some kind of abrasive force, we have a stratified squamous, where we're not, we can then switch back to that respiratory type epithelium so that we can have those protective mechanisms like that mucociliary escalator acting. So this is kind of a cross section. So at the top here, we have a false vocal fold. We then have this sort of inlet here called a ventricle that sort of separates. You can see there's quite a clear delineation between the false vocal fold, which has got lots of glands and it's sort of subendothelial tissue. And then as we get to the true vocal folds, I don't know why, if you were going to make a textbook, you'd have the best picture you could. This is a picture in the textbook and they go, oh, by the way, part of the vocal cord is missing. You couldn't have found an intact one, could you? So the outline is where the true vocal fold was. I couldn't find a better one. And that's where we see that stratified squamous epithelium. You'll see a lack of glands here, but what we see here is our vocalis muscle and ligament, so involved in sort of stretching and pulling the vocal folds to fine tune the sound that's being produced. You can't really see the epithelium very clearly on there, but if you zoom in at the top, there's the epithelium of the false vocal fold, and there's the epithelium of the true vocal fold. You'd be, forgiven for thinking they're the other way around, because normally when we talk about stratified squamous epithelium, we imagine a really chunky one, right? Like when we looked at the esophagus, it's like 15 to 20 cell layers thick. It's not always that thick. So here, we might only see something in the region of like 5 to 10 cells thick for the stratified squamous epithelium. I remember a squamous cell is a very flattened cell. So when we have 5 to 10 of those, we can still end up with ultimately a very narrow looking epithelium. So even though at first glance you might think, right, that's the stratified one because it looks chunkier, it's not. This is the stratified squamous epithelium, 5 to 10 cell layers thick. This is that pseudo-stratified columnar epithelium, that respiratory type epithelium. So sometimes you can't just take a glance at it and assume which cell type is which epithelium is which. Moving down then into our trachea, so this is a cross section straight through. We can see what we've got here in the trachea is this C-shaped ring of cartilage. So this sits, if that's the letter C, it sits with sort of the open part of the letter C facing posteriorly in the neck. And then behind that, behind the trachea, we'd find our esophagus. So we have this cartilage ring. So their job is to give the trachea some rigidity, because as I said, we don't want the only passageway to get air into our body to ever collapse, so it keeps it nice and open. At the back then, where that C-shape of cartilage is not connected, we have this trachealis muscle. So by having this sort of incomplete ring of cartilage, we still do have some degree of kind of compression that can occur, or sort of narrowing of the airway diameter. So for example, when you're coughing and need to kind of increase the pressure, the airway is narrowed to increase sort of the speed, the velocity of that air exiting to help get whatever it is out. We can also see some slight expansion of the trachea as well during inspiration. So there is kind of some flexibility for it to sort of change its diameter, but it stays open because of those cartilage rings. If we look then, we can see it's got a respiratory type epithelium. So again, that's pseudo-stratified ciliated columnar epithelium with goblet cells. And you can see, well, you can't actually see on this because it's not zoomed in enough, but we have lots of basal cells, particularly more in the upper trachea. So again, that's just because it's more likely that the upper trachea will be exposed to sort of injury. So for example, through smoking or something like that, it's the upper airways that are going to come into contact that more frequently or more likely. So they have more basal cells to help with the renewal of the epithelium to try and make sure it stays sort of intact and healthy. And we have, again, just like we saw before, seromucous glands here in the submucosa to again help with secretion of sort of the mucus and that watery serous secretion as well. But then as we sort of go down through the trachea, we see less in terms of those seromucous glands in the submucosa. So we're in the trachea, we've got those lovely rings of cartilage. We then see the trachea bifurcate or split in two. And where they split, we have this region here called the carina. The mucosa at the carina is particularly sensitive. So if something's inhaled into the airways and it tickles that bit of the mucosa, that can trigger the really strong sort of cough reflex. So that particular bit of mucosa is sort of highly got a lot of like receptors and things to sort of detect if anything's come into the airways, then trigger that cough reflex. So we have the trachea bifurcating or splitting in two to our main or primary bronchi. So one for the right lung, one for the left lung. The primary bronchi then splits into secondary or lobar bronchi. So we have 1, 2, 3 on the right for the 3 lobes on the right, and on the left we have 2 of those for the 2 lobes on the left. And then those lobar bronchi, so feeding the different lobes of the lung, will then branch into tertiary or segmental bronchi, feeding the different bronchopulmonary segments of each lobe. So each lobe of the lung also has these kind of structural divisions called bronchopulmonary segments, which are fed then by these tertiary bronchi. And you can see with each division, as we go down, the diameter of the airway decreases. So the trachea has a diameter of around 2 to 2.5 centimeters. But by the time we get down to those tertiary or segmental bronchi, we're looking at a diameter of about 0.1 to 0.6 centimeters. The obvious thing to bear in mind, though, is we have one trachea and we have many of these. So even though the diameter is decreasing, we're not decreasing the capacity for like airflow because we've got lots of them, okay? So as we go down through the trachea into the bronchi, we don't have C-shaped rings of cartilage anymore. As we move into the bronchi, they turn into kind of irregular plates of cartilage. And as we keep going down and down and down, when we get to sort of the bronchioles, we'll see that they then don't have those cartilage in. So we start with those quite sort of substantial C-shaped rings, sort of irregular, sort of overlapping plates, and then it diminishes as it goes down to the bronchioles. So this is our main bronchus. So again, a respiratory type epithelium. So this is a nice bit. We're not seeing switching between different epithelial types. It's a respiratory type epithelium. We do start to see little changes, fewer goblet cells. Maybe they start to become a little bit shorter as we go through. Although you can't really see it on here because we typically need a special stain to see elastin, we have elastic fibers, as we mentioned before, in the walls of the sort of bronchial tree as it goes down. Again, so that during that passive exhalation process, the recoil of those elastic fibers can help reduce the size of the sort of respiratory system to help expel air. We have between the mucosa up here and then the submucosa here, we have these sort of smooth muscle fibers. Where we've seen this kind of positioning of those muscle fibers in sort of the wall of a tissue before, so when we looked at this in the GI tract, they referred to it there as a muscularis mucosa. We don't tend to use that term here. Call them muscle fibers. We don't give it that name, even though it's kind of situated in sort of a similar position in terms of the layers of the wall. Muscularis mucosa tends to be a sort of term that's sort of uniquely applied to the GI tract. But it's kind of in the same location. It's between the mucosa and the submucosa sort of layers. We have some of these muscle fibers. In the main bronchus, not quite as substantial in regards to the overall proportion of the sort of wall. Doesn't represent a high proportion of the tissue there, but as we descend through the bronchial tree, we'll see more muscle fibers, and proportionally they'll make up more of the actual respiratory tree wall as we go down through the bronchial tree. We see fewer seromucous glands here in the submucosa, so sort of diminishing seromucous glands as we go down. And as I mentioned, we then see, you can see a C down there, so that's some of the cartilage, but it goes from those C-shaped rings in the trachea to these sort of irregular but sort of overlapping plates of cartilage as we go down. So still providing some rigidity to the bronchi. And as we'll see later, as that cartilage decreases and we see proportionally more smooth muscle, that's then where the lower airways can have that sort of collapsibility. So that was a main bronchi. So remember, we go main or primary bronchi, then we have lobar or secondary bronchi, and then we get to our tertiary or segmental bronchi. So again, we still see a respiratory type epithelium, but that starts to transition to a ciliated simple columnar epithelium. So it doesn't quite look like that pseudo-stratified version anymore, just looks straight up like a simple epithelium. It's not tricking us anymore in its appearance. Again, we're seeing fewer serum mucus glands in the submucosa as we go down. We see sort of relative to the overall thickness of the airway, we see proportionally thicker smooth muscle. So that's labeled here. So this stain in particular stains muscle. It's not the best stain, but stains it kind of an orangey color. So you can see compared to the last one where we had a few sort of piddly fibers of muscle, we now see a much thicker sort of layer of muscle. Again, you can see bits of the cartilage here, but they're kind of irregular plates of cartilage. So we don't see, like we saw with the trachea, we saw a C shape all the way around. We've just got little pieces, little plates of cartilage surrounding the tertiary or segmental bronchi. From the bronchi then, further divisions, we term them bronchioles. So bronchioles are those airways that are less than 1 millimeter in diameter. We go from that ciliated simple columna, the cells start to become a little bit more squat, they become more cuboidal. We see few goblet cells here, so we've transitioned largely away from that kind of respiratory type epithelium now. We have no submucosal glands, so we can't really see any of those glandular structures and that sort of subendothelial tissue here. But what we do see, labeled M here, is quite a thick, smooth muscle layer. So again, the smooth muscle layer, it's, helps with regulation of airflow into and out of the respiratory system. But crucially, in the bronchioles, we have no cartilage. So if the airway is irritated, so for example, in asthma, that smooth muscle can inappropriately contract, close off the lumen, close off the airway and prevent airflow. So it's obviously there in health for a reason with regulation of airflow, but it can be inappropriately stimulated to cause bronchoconstriction. As we'll see next week, that's not the only issue we have in asthma. We also have lots of mucus in the airways and other things that are preventing that airflow as well. But that bronchoconstriction is one of the key features. We see that sort of collapse of the bronchioles in that case. In terms of when we get to sort of the lower bronchioles, we end with what's known as terminal bronchioles, which then branch into respiratory bronchioles. And the reason we give them two different names is because the terminal bronchioles represent the last part of the conducting zone. So everything up until this point has just been about transmitting the air from the outside down into the respiratory zone. And then our respiratory zone starts with our respiratory bronchioles and extends to include all of, obviously, our alveoli as well. So the respiratory bronchioles are included because they'll have sort of alveoli branching off the walls of the bronchioles. So we are seeing gas exchange taking place at these structures as well. This isn't the best diagram. Another one in a minute that shows you the kind of alveoli branching from those respiratory bronchioles. But the respiratory bronchioles down, that is where we see our gas exchange taking place. And as I said, as we transition down, those cells have become sort of more squat. They're now cuboidal. They're still ciliated in terms of the terminal respiratory bronchioles. But when we get to the alveoli in a moment, we'll see them transition again into a different type of epithelium. Because we've seen a reduction in goblet cells, we've moved away from that respiratory type epithelium. We don't have goblet cells anymore. This is then, I don't know why I've called it Clara there. I should have changed that. Apologies. Club cells here. Replacing those goblet cells to providing that kind of respiratory protection in absence now of that mucociliary escalator, which we don't have anymore. So we have a different protective mechanism, so that can help with sort of detoxification things. And as we'll see in a moment, we also have cells in the alveoli called alveolar macrophages, so a type of immune cell that can engulf pathogens as well. So where we don't have a mucociliary escalator, we have other protective mechanisms to protect those delicate respiratory surfaces. So this is quite a cool image. It shows you the sort of terminal bronchi here branching into the respiratory bronchioles, where you can see all these kind of like bowl-shaped structures stuck to the side of this respiratory bronchiole. That's alveoli. So that's why the respiratory bronchiole is part of the respiratory zone, because there's little alveoli sort of stuck or openings from the side of the respiratory bronchiole. And then we go down to the sort of alveoli here, so these kind of clusters of air sacs where we see sort of the majority of our, or where we see our gas exchange taking place. So you can see terminal bronchiole up here, respiratory bronchiole, some little alveoli protruding off the side, but those respiratory bronchioles will then branch to form these alveolar ducts, which have alveoli branching off either side, and then these alveolar sort of sac structures here. So this is where we see all of that gas exchange taking place. And this is what it looks like histologically. So even though it looks like a lot of empty space, that's what it looks like. There are lots of millions of tiny little balloons, essentially. They're not balloons, but that's what they kind of would look like. So here we can see sort of that would be a cross section of our terminal bronchiole, then branching into our respiratory bronchioles. We have these what's called alveolar rings. So these are sort of denoting where you see branching from the respiratory bronchiole in then to our alveolar ducts. And then we have our alveolar sacs where several alveoli kind of open into the same region. So this is kind of looks funny because it almost looks like a tissue that's kind of been destroyed a bit, but that's what it looks like. And all of these surfaces here, all of these walls of the alveoli, within those walls we'll find capillaries, so really small blood vessels for gas exchange. So this is why, you know, even though it looks very thin and it is very delicate, it needs to be very thin to provide efficient gas exchange. We wouldn't want to have a stratified epithelium forming the wall of our respiratory membrane because that's just a massive barrier for diffusion of gases. We want as thin as possible alveolar wall for efficient gas exchange. So our alveoli are forming the respiratory membrane. We've got this dense capillary network sort of surrounding them in the intraalveolar septa, so in the connective tissue, to make sure there is really good contact between, you know, the air coming in and the blood vessels for that gas to move, or the oxygen to move into the blood and the carbon dioxide to leave the blood. And to make sure that we have efficient gas exchange, we have sort of mechanisms built in that if one of the bronchioles, respiratory bronchioles, was blocked, the alveoli being fed by that respiratory bronchiole can still receive air by another route. So it's collateral ventilation. So if one bit goes down, hopefully those alveoli can still be used in gas exchange because they can get air from somewhere else. So we've got, here's our sort of respiratory bronchioles. We've got these channels of Martin. So you can see passageways connecting one bronchiole to the other. So if there was a blockage, you know, say somewhere down here on this one, this alveoli could still get air via another route. We've got channels of Lambert. So this is between bronchioles and directly with the alveoli. And then we have pores of Cohen here. So pores or channels directly between alveoli. So a way by which if there's some blockage or some impairment of, you know, passage of air down this route, this alveoli can still receive air and still participate in gas exchange through these other channels, through these other routes as well. Good, right? We want that to take place, but where do you think that could be of detriment to us?
Infection.
Yeah, say more.
Giving infection easier and wider access.
Yeah. So say we had some bacteria down this alveoli, well, we've got now like an open passage for them to get into this alveoli and for them to spread. So it's great in terms of making sure we can use the respiratory membrane as much as we can for gas exchange, but it provides the opportunity for infections to spread more easily because we've got all these little pathways for the bacteria to move around and spread. So cartoony diagram to start with. So this is one alveolar airspace. There'll be another airspace here, another airspace here, another airspace there. So you can see we've got two quite distinct types of cell forming the wall of the alveoli. We've got these very flattened out cells, which are type 1 pneumocytes. So the word pneumocyte is just referring to the epithelial cells of the alveoli. So type 1 pneumocytes, as you can see in the diagram, are simple squamous. So you can see a sort of oval, flattened nuclei, and then very, very little cytoplasm that's spread very sort of thinly out. So very sort of elongated cell, but creates a very, very thin kind of cell width for movement of gases across it. And then we have a second type of cell here forming part of the alveolar wall, which is our type 2 pneumocytes. And these are more cuboidal. They don't quite look like a square like you put up in the cartoon diagrams, but you know, their height and width are roughly the same. So they're cuboidal cells, and they produce that surfactant that was mentioned earlier that I'll discuss in a moment. So the type 1 pneumocytes, they account for around 40% of the pneumocytes in the alveoli, so making up the alveolar surface, but they comprise about 90% of the surface area because they're long, big, sort of flattened out cells. So even though there's less of them, they sort of comprise more of that wall structure. Whereas the type 2 pneumocytes, even though there's more of those in number, so they account for around 60% of the pneumocyte population, they comprise only about 10% of the alveolar wall. So the majority of the alveolar wall, which is where the gas exchange is taking place, is those type 1 pneumocytes. But those type 2 pneumocytes are numerous because they're really, really important. So this is what it looks like histologically. So you can see the type 1 pneumocyte, very, very sort of scanty, very little cytoplasm. It's just creating enough of a wall that it's kind of intact, but it wants to be as thin as possible for efficient movement of gases across the alveolar wall. Whereas the type 2 pneumocytes are a little bit chunkier, and they're chunkier because they have more machinery for manufacturing the surfactant. So they'll have more mitochondria, they'll have more endoplasmic reticulum because they're actually making something. So this is one of our, again, our type 2 pneumocytes shown here. So they have very large nuclei compared to the sort of very thin, flattened nuclei of the type 1 pneumocytes. Within that nuclei, the genetic information, the chromatin content within there is very fine and granular and evenly dispersed. So it's not clumped together in any way or sort of all located at the periphery. It's kind of all dispersed throughout the nuclei. So it gives it this sort of evenly dispersed staining pattern. And although it's not very obvious in this one, we sometimes see prominent nucleoli. So nucleoli is where we have manufacture of our ribosomes. So when we have a very active cell in terms of producing a product, we need lots of ribosomes where we will then see protein manufacture. It's got lots of cytoplasm, and in that cytoplasm, we see lots of mitochondria, lots of rough and smooth endoplasmic reticulum. That's because they're little machines for making surfactant. So surfactant is a mix of phospholipids, carbohydrates, and protein. They aren't the only cells that make this. So the club cells do produce some components of surfactant, but the type 2 pneumocytes are the main ones that are making our surfactant. And although we can't see on this, they will have apical microvilli just to enhance their secretion of surfactant as well. So a bigger surface area so they can be secreting more of that surfactant. And as it was highlighted before, surfactant is really, really important because it reduces the work of breathing. So if our alveoli were simply lined with water, water molecules have very strong attractive forces for each other. So if I'm a water molecule, I'm grabbing that one there, I'm grabbing that one there, I'm pulling it towards me, and that's going to cause collapse of the alveoli. Or it's going to just make it really provide more work for me to overcome those sort of attractive forces for me to enlarge the alveoli to breathe. So what surfactant does is it disrupts those attractive forces between water molecules to reduce surface tension, so reduce the sort of pull of those molecules to want to pull inwards to help prevent collapse of the alveoli and to help ensure that expanding them is easier for us to do, okay? This is really, really important that we have surfactant when we take our first breath. So babies who are born too early, before around, I think it's around 34 weeks, might not have enough type 2 pneumocytes. They might not be developed enough yet. They might not be secreting surfactant, and that infant won't be able to overcome those attractive forces to inflate their lungs. So they go into infant respiratory distress syndrome. They have to be artificially ventilated and they have to be given artificial surfactant until their body's had enough time for those type 2 pneumocytes to mature and start making their own surfactant. We also mentioned, and again, this will make a lot more sense when you talk about the process of ventilation and moving air in and out of the body, but in the walls of our alveoli, we have elastin fibers. So here's an elastic stain. The elastic fibers are staining black. So this provides recoil during expiration. So as I said, during inspiration, the alveoli are going to be through a series of processes, but let's just say they're pulled open by the movement of the chest wall. bit more complex than that, but it's through the contraction of the chest wall muscles that ultimately the alveoli will increase in size and air is drawn in. And then as they're kind of pulled open, what happens is the elastic fibers are stretched, they're put into tension. So we have these opposing forces of kind of the muscles wanting to move the respiratory sort of alveoli, open them up, but opposing that are those stretched elastic fibers want to pull it back in. So as we reach sort of maximum inhalation, that's where we see a balance between the force pulling the alveoli open and the elastic fibers wanting to pull the alveoli in. And then when we release the contraction of the muscles, there's nothing then to overcome those elastic fibers, and they will then return to their original size, return the alveoli to a smaller size, and air flows out. So I think it's exactly when you go through that process of what actually happens in terms of the pressure changes and all that kind of stuff. But that's why the elastin fibers are really important, because exhalation is largely, at least quiet sort of exhalation, is a passive process. These provide the recoil to then reduce the size of the alveoli. So as the alveoli are expanding through muscular force, they increase in size, and then it's the elastic fiber recoil that then reduces their size for expiration or exhalation. We mentioned that by the time we get down to level the alveoli, we don't have those goblet cells, we don't have the mucociliary escalator. We do have the club cells, which can help in sort of detoxification of harmful substances. We also have alveolar macrophages, so macrophages resident in the lungs, so they're called alveolar macrophages, sometimes called dust cells, which I think is quite a cute name for them, but not always the most helpful name. And they are phagocytic cells, so they can engulf debris, particulate matter, organisms that might get all the way down to the alveoli. Hopefully all those are the systems and measures we have in place stop anything getting all the way down, but if it does get to the level of the alveoli, we have our macrophages. So they phagocytose and inhale bacteria and particulate matter. They can also remove superfluous surfactant, so just any extra surfactant that is not needed. They also release chemical signals to help recruit to other phagocytic cells, such as neutrophils and other immune cells, to help sort of address, if there is, bacteria there, help address that. You can see them on this. So again, this is a sort of immunohistochemistry. So where again you see these brown blobs, that's our macrophages staining up there. So they've looked for a particular marker on the surface of the cell to highlight them. So all of these brown blobs in the alveolar wall, that's our alveolar macrophages. Once they've engulfed something, they are motile. The cells are motile, so they can move up the respiratory system a little bit and be captured by that mucociliary escalator and then be taken up to the pharynx and swallowed or spat out. They could move into the lymphatic system and enter from there a lymphoid tissue where we have the B and T lymphocytes who can look at what they've captured, see if we need to raise an immune response against that. Or they could end up staying where they are in the lungs. Now, obviously, that's not what we'd want to happen, but if they are overloaded, so a thing that alveolar macrophages sort of engulf a lot of is carbon particles, so from exhaust fumes, for example, from cigarette smoke. And if they're overwhelmed, if they've engulfed just loads of these sort of harmful carbon particles, that kind of damages their function and they can't move anymore. They're not motile and they end up getting sort of stuck in the septa of the alveoli. And because we said they're important for directing immune response, that can cause prolonged inflammation, which can cause fibrosis and scarring and reduction of lung function. So, cigarette smoke can really damage alveolar macrophages. They're doing their job, or as best they can, but then the body's not able to clear them out, and that can be sort of detrimental along with all the chemicals and things in cigarette smoke. So this is a particularly stark example, not from a smoker, but from a coal worker. So here we can see all those black dots there, all that sort of black masses there, all the macrophages that have engulfed the carbon particles that individual's been inhaling. And you can see, if we go back to the previous diagram, you can see even though there's macrophages there, we've still got a nice thin alveolar wall. We want it nice and thin for efficient gas exchange, but where we then have a prolonged immune inflammatory response, we get fibrosis, we get scarring, we get thickening of the alveolar walls, which we obviously don't want because that's going to make it so much harder for gas exchange to take place. So that individual's not going to be you know, their body's not going to be receiving oxygen as efficiently as it should. They're not going to be getting rid of carbon dioxide as efficiently as they should. So it's really important that the alveolar wall is healthy. It's not prompted through any inflammatory process to, you know, sort of thicken. In health, the air-blood barrier, so that distance that gases have to pass from, you know, the air in our alveoli into the blood is, you know, 100 times thinner than tracing paper. It's incredibly thin. So it's really, you know, it's really efficient in terms of gas exchange. So as I said, the gas exchange surface is the type 1 pneumocytes. So we have in our alveolus, here's sort of part of one of those type 1 pneumocytes. So our squamous cell with a flattened nucleus, and then just a really thin cytoplasm here. If you recall, our capillaries, our blood vessels are lined by, we call them endothelial cells, but they are a simple squamous epithelium as well. So here's an endothelial cell, but it's also a simple squamous epithelium. These cells will sit on a basement membrane. So the cells of the alveoli will sit on a basement membrane. The cells of the endothelial cells will sit on a basement membrane. But what happens at the air-blood barrier here is those basement membranes fuse to make that distance even thinner. So we have an incredibly short distance for oxygen to diffuse into the blood, carbon dioxide to diffuse out. So it's less than 1 micrometer. So it's really tiny distance. And those gases are lipid soluble as well. So they can just move straight through the cell membranes of the simple squamous and of the alveolar type 1 pneumocytes and the endothelial cells. So super thin respiratory membrane there for really efficient movement of gases. And we can see that on a scanning electron micrograph here. So here we've got a type 1 pneumocyte. So looks more to be the nucleus up here, but here's just a thin cytoplasm. Here's a cytoplasm of the endothelial cell. They're fused basement membranes. You can tell the scale we're looking at here because that's a red blood cell down there. So we're really zoomed in quite a lot to see this, and you can see it's an incredibly thin barrier for diffusion of gases. So anything that's going to damage this or affect it is going to have a substantial impact on the efficient respiration. That's it. Some good textbooks there. The top 2 are kind of like the Bibles of histology. So if you ever want to do some further reading, those top 2 are what I'd recommend, but these ones also have some good info and pictures as well. Thank you.
Daniel, merci.